Hi guys, Dr. Orlando here with a video on DNA replication. So why does a cell decide to replicate its DNA? Well, a signal would go to that cell to tell it to do so, and it would get that signal if it needs to go through mitosis or meiosis, so cell division. So we do this to prepare for cell division. All right, if you want to make a copy of yourself, cell division, then you have to copy the DNA. Okay, so this is in preparation for cell division. So we start with the DNA, which looks like this. Okay, it is double-stranded, double helix DNA. It will be loose in a chromatin form, and then it will be copied. We call this the original DNA. We call it the parent strands. And so there are two parent strands here in the DNA. When it's all said and done, the result is we will have two exact copies, two DNA molecules, and they will be exact copies. Okay, but they won't be. They will be made up of parent strands and daughter strands. So you'll have each DNA molecule will have one parent strand shown in black and one daughter strand shown in pink. So we have a little bit of old DNA mixed with new DNA. Not new information, but a new copy of the DNA. Okay, so we have conserved some of the old DNA. And we call this semi-conservative replication. Semi-conservative replication. Old and new together. So for replication to occur, DNA replication to occur, it requires many enzymes, and I'm just going to cover a few of them here in my description. And I want you to make sure that you know these and what their functions are and how they assist in replication. So helicase, and I've drawn a little key here, so when I actually draw the picture with replication, I don't have to label everything. Okay, so helicase is going to be my little blue square, and its job is to unzip the DNA. So it's going to separate the strands, separate the parent DNA strands. Primase is going to lay down an RNA primer. And this is a short segment of RNA, and I like to say it's like bait. It's like bait for DNA polymerase. So that DNA polymerase knows where to start. Okay. So DNA polymerase is the one that will find the primer in black. Find the RNA primer and will build the new strand, not just the new strand, let's say the daughter strand, so we're using the right terminology here. We'll build the daughter strand one nucleotide at a time. Thankfully, we'll see, and it won't be easy for me to draw this. I probably won't actually draw multiple DNA polymerases, but imagine that really long strand of DNA and lots of DNA polymerases working on different segments, but each DNA polymerase is only adding one nucleotide at a time. Okay, and then finally, ligase, we'll see that on one side, and that side is called the discontinuous synthesis side, or the strand is called the, we'll write it this way, the lagging strand, the lagging daughter strand. Will be made in segments. So ligase will bind the DNA segments together. The lagging daughter strand will be made in segments, so the ligase will bind the DNA segments together. Think of to ligate something means to tie it off or tie it together, so we're going to ligate those together. All right, now that we have all of our enzymes figured out, we'll go ahead and look at how replication occurs. So here is a picture of the parent DNA that we're going to copy, and I blocked off a little segment here and say we're going to zoom in on that, and then we see this. Okay, and what happened here is it began to open up, and it formed a bubble. So we call this the replication bubble. And imagine this long, long strand of DNA. All along it, we have these replication bubbles opening up. Well, who's opening it up? Who did I say unzips the DNA? Who unzips the DNA indeed? This guy. Helicase. So helicase is going to move along, unzipping our DNA. Okay? All right. And it's doing that all along the way, making lots of little replication bubbles. The replication bubble that you see here, it does have these, actually right where my helicase is pointing, these kind of areas that aren't quite open yet or will be opened. We call that the replication fork. Okay, so before we go any further, I want you guys just to practice base pairing by looking at this strand of DNA that I have opened up. I'm going to move it over here. And I'm going to tell you that this is the 3' carbon here. Okay? And this is a 5' carbon here. So this strand, this parent strand, runs 3' to 5', and this strand, because we're gonna work in this direction, this strand is going to run in the 5' to 3' direction. Okay, and we are going to copy in that direction. Okay. 3' down here, and 5' right there. Okay, so what we need to do is complement our other side here. So I'm going to go ahead and fill in the letters, and as I go along, you can pause it if you want to write out the code, or you can just follow along. If you want to pause it, pause now, because I'm going to write them in. Okay, so we know that A pairs with T, G pairs with C, T with A. Okay, so those were hydrogen bonded together to keep it nice and tight, but helicase came along and unzipped them. Undo those drawings there. So now they're nice and open so that DNA polymerase can come in and start making copies. But first what has to happen is primase, I don't remember what color I gave him, primase, which is red, okay, he's got to come in and lay down a primer. Okay, so I'm going to make a really short primer here. My primer is only going to be 2 nucleotides long, so let's draw our primerase in here. Okay, and he comes in and he brings in a couple of nucleotides. And I know I put a U there, we haven't talked about that, but RNA doesn't have any T's, it has U's instead, so A is always going to pair with U. So this is my RNA primer on this side. Okay, that way DNA polymerase, I'll draw him in now, knows where to attach. So we're going to put our DNA polymerase in here, this nice big pink rectangle. Now, a rule for DNA polymerase, big time rule. Let's move it over here and write this rule because it's really important. DNA polymerase. Whoop. It has a way it likes to work. The only way it will work, the only way this DNA polymerase will work is in the 5' to 3' direction. That's the only way it will build. The new strand will be built 5' to 3'. It's like a guy with a contract. It's the only way that he's going to work. So that's good, because this first strand that we're looking at, remember, it's got we're starting on the parent strand starts with 3', therefore the growing strand will start with a 5', right? That's good. So when we build it, this 5' is going to go to a 3', which is exactly the way DNA polymerase likes to work. It likes to just keep adding on in the 5' to 3' direction. So let's look at that. Let me undo that so I can draw. So let's go ahead. I'm going to use, color should I use for growing strand? Let's use blue for our growing strand of DNA. So now we're going to add a new strand of DNA here. Okay, so I'm going to put my backbone in there just to make it a little bit easier. But you do need to remember that it is added 1 nucleotide at a time. Okay, so each little section here, if I were to circle this, now that right there, that's a nucleotide. Okay, and if we were to zoom in on it, we'd see that this part right here is where you find the sugar and the phosphate. This is the nitrogenous base. Okay, so that's 1 nucleotide. And it's going to, that's how DNA polymerase is going to do. It's going to add 1 nucleotide at a time, moving in the 5' to 3' direction. Okay, so I'm going to move right down the line, adding my nucleotides. And as I add the nucleotides, that's indicated by me adding the base that belongs there. Oop, looks like I'm missing a line there. Okay. That got a little messy here. I want to fix him. There we go. Okay, so we just complemented that side. Okay, and DNA polymerase is going to continue down this side. It's going to keep going down this side until as long as helicase continues to unzip, as this guy continues to unzip, DNA polymerase will keep moving on down the line adding its, we can draw DNA polymerase moving down the, oops, wrong color. DNA polymerase is pink in our picture. So it's going to keep moving down the line, adding nucleotides in the 5' to 3' direction. Okay? So at the end of this strand, we have a 3', and at the beginning, we had the 5'. So 5' to 3' direction. The brand new strand built just the way DNA polymerase likes to work. And he's going to keep going along as long as helicase unzips, or and until he runs into another DNA polymerase, okay, or runs into another DNA polymerase. He's just going to keep going until something gets in his way, okay, and then eventually this entire strand will be built. So you can see that this process works in a continuous fashion. So we say that this side is an example of continuous synthesis. Okay, continuous DNA synthesis. And that this strand, because it goes more quickly, it's easier, it is called the parent strand there. I shouldn't say, not the parent strand isn't called that. The new, let's point to the right strand. The new strand that's being built is called the leading strand, because he's going to go fast, because he works in the direction DNA polymerase likes to work. Okay, on to the other side. So now we got to go, all right, so if DNA polymerase has this rule that he refuses to work in any direction except for the 5' to 3' direction, well, let's look at this what I'm going to call the bottom strand here, and see what's going on with him. So this strand runs 5' to 3'. That's how the strand runs. So imagine that's how the parent strand runs. Imagine what the daughter strand is going to look like. It's going to start with a 3', right? It's going to start with a 3' here, and it's going to go down. It's going to start with a, let's see if I can zoom in so we can draw this. Have more space to draw. Okay. It's going to start with a 3' on this end and work to a 5'. Well, that's not going to work for DNA polymerase to slide in that direction, because he refuses. He wants to go 5' to 3'. So how does it all work out? Well, let's see. Let me clean up my space a little bit here so we can have more room to draw in our other strand. Okay. All right, so, well, the first thing that has to happen, we do need RNA primerase to come in and lay down a primer for us. But he's going to put his primer, we're going to put our primerase working right here. I'm going to put it right there. And it's my red. Okay, so we're going to go U. Remember, as I said, RNA is a little bit different, doesn't have T's, it's got U's, so we have to put a U there. And that's our primer. Okay, that's our RNA primer. So now DNA polymerase says, okay, that's fine, I like RNA primer, that's my bait, that's what I am attracted to, it's why I go start doing my job. So he goes and he finds oh my goodness, I'm doing too much. Okay, let's get that. Let's find pink. There we go. So he finds that primer, because like I said, it's like bait for DNA polymerase. Can't resist it, has to attach. And what direction did I say this guy has to work? He has to build 5' to 3'. And where was the 5'? The 5' is here. The 3' is here. So he has to build in this direction instead. All right, so let's go ahead and make DNA polymerase work in that direction. So go ahead, we're going to add the bases in that direction and see what we come up with. Let's draw our backbone first so that we have something to draw it onto. Okay. Okay, I had to move my strand a little bit to make room for how I'm drawing it here. All right, now we are going to add our bases. Okay, so C is going to pair with G, C, T, G, A. And so RNA polymerase is going to keep just moving there in that 5' to 3' direction. Moving on down the line, adding the bases. So what happens when he gets to the end? Well, if it's the end of the strand, he'll be removed from that strand, right? But eventually he's gonna stop, right? He's gonna hit some space that has already been done, or he's gonna hit the end of the strand, and he can't go any further. So he's gonna come off, and he's gonna go back and find another primer. Okay, so let's draw another primer in here. Let's put another primer right here. There's our primerase. He's going to add a primer, and it's going to be okay, like that. Okay. So then polymerase says, okay, I can come back here. I found a primer. I'm going to go ahead and start doing my job. So he's going to start adding nucleotides to this growing strand. Just like that. Okay? All right, so now what do we see there? I'm going to erase this guy, this 5' in my way. Get rid of him. So now what do we see? Well, we have fragments. We don't have continuous synthesis. We have fragments. Okay, so this strand, or this type of synthesis, let's do that first. This type of synthesis is called discontinuous synthesis. And this strand, because it's going to be slower to produce, since DNA has to jump off and start again, that's going to be called the lagging strand. Okay. The daughter strand that is built in this direction, in this way, is called the lagging strand. So, something else that has to happen here. When you look at this, it doesn't look quite right. It doesn't look finished, right? It's not. We end up with these little fragments. And these fragments, boy, this is getting tight here. Okay. The fragments, I'm going to circle it. See that fragment right there? That fragment is called an Okazaki fragment. I'm going to undo it just so that you can see it. I'll go like that. It's called an Okazaki fragment. Okay, so fragments of DNA that are built by discontinuous synthesis, and they needed to be joined together. Well, I said that there was an enzyme that we have whose job is to join fragments. His name is ligase. So we'll put ligase in here, and ligase is going to join like that. Ligase is going to bring those strands together. And I'll ligate the strands, bring them together. Okay, so there, now we have replicated our DNA, and you can see that at least this little segment that's replicated, that it was semi-conservative, that each growing strand of DNA is made up of parental DNA and daughter DNA, parental DNA. When it's all said and done, we will have those two separate strands, just like we see here, the result. Okay. And as the, I didn't mention this, but as the nucleotides are being added to the growing strand and it gets longer and longer, the hydrogen bonds naturally form between the bases, and the hydrogen bonds that create the helical structure begin to form. So it is being copied, being built, and being twisted all in the same, within, the same time frame. It doesn't all get built and then suddenly something twists it. It's being a continuous process that builds and twists at the same time. Okay. When it's all said and done, these two DNA molecules will be absolutely identical. So I know we talked about the cell cycle a little bit, so I can put this in there for you, that initially that DNA would be a single, not like that, would be one single chromosome, looking like that with its centromere there, all right? And then when it goes through replication, you end up with two exact copies. They are joined to each other with those centromeres. Okay. And then we call these guys, this a duplicated, or replicated, either one, duplicated chromosome. Okay. And that these two gals here, they are sister chromatids, and they are identical to each other. So as soon as it's done being made, these two attach to each other. It's a good way to keep everything organized in the cell, so when it comes time to split them later, they are where they need to be. All right, if you have any questions on DNA synthesis, please let me know. Have a good day.